Today, we're going to examine a novel bioelectronic platform that makes it possible to implant electronic devices into the brain without surgery. This technology, which the researchers call Circulatronics, uses cell electronics hybrids to achieve autonomous delivery and highly focused neuromodulation. The work was recently detailed by Yadav and colleagues in Nature Biotechnology. So here's how we'll break down our analysis. We'll start with the foundational concept and the problems it's trying to solve. Then we'll get into the specific engineering of the electronic devices themselves. After that, we'll look at the biological strategy they used for autonomous delivery, review the in vivo data on stimulation and precision, go through the safety profile, and then wrap up with the potential future of this work. All right, first, let's set the stage and establish the specific problem this research is aiming to solve. Right now, the standard for bioelectronic brain interfaces is pretty invasive. It requires intracranial surgery, which, while it can be effective, comes with a set of inherent risks. Things like tissue damage from the implant itself, the potential for infection, and other surgical morbidities. So, this work explores a completely different pathway. What if we could adapt the intravenous delivery route, which is standard for drugs, for the administration of electronic devices? The platform they've developed is called Circulatronics, and the core idea here is to completely decouple the implantation process from manual surgery. Instead, tiny, micro-scale electronic devices are introduced into the bloodstream and are then guided to a very specific target tissue by living cells that act as their own autonomous biological delivery system. Now, to make this idea a reality, there were three major technical challenges that had to be overcome. First, they needed to fabricate wireless electronic devices that were small enough to actually navigate the microvasculature without causing a blockage. Second, they needed a strategy to prevent these devices from being rapidly cleared from the bloodstream. And third, they needed a mechanism for the devices to recognize and then stably implant themselves within a precise, predetermined region of the brain. Okay, so let's dig into the engineering of the core electronic component of this system. These devices are called subcellular-sized wireless electronic devices, or SWEDs. Essentially, they are free-floating photovoltaic microparticles. A really critical design feature is their diameter. It has to be 10 micrometers or less. As you can see, this is smaller than key circulating immune cells like monocytes, which is a feature designed specifically to allow them to pass through tiny capillaries and prevent any kind of embolism. The SWEDs themselves are fabricated as a 3-layer stack of functional materials. If we look at the table, the structure is made up of a transparent, biocompatible anode of PDO-TPSS, a biocompatible titanium cathode, and then sandwiched in between is an organic semiconductor active layer. This active layer generates a current when it's stimulated with light. And of course, these materials were all selected for both their electronic properties and their known biocompatibility. A pretty significant engineering achievement here is the development of a wafer-scale fabrication process. This makes it possible to produce very large quantities of these devices with consistent morphology, which you can see here in the SEM image. This kind of scalability is absolutely essential for extensive preclinical testing and, of course, for any potential translation of the technology down the road. Of course, a fundamental question is whether you can even power these devices through biological tissue. This ex vivo experiment addressed that head-on. The chart shows that these 10 micrometer SWEDs can generate about half a nanowatt of power when they're illuminated with near-infrared light that's transmitted through an entire mouse brain, even with the skulls still intact. This was a key finding that confirmed the feasibility of transcranial wireless power delivery. Another interesting feature is that by using different organic semiconductor blends, you can achieve functional multiplexing. As the data here illustrates, one blend has a peak response at 520 nanometers, while the other responds at 785 nanometers. This spectral difference makes it possible to independently address and control different populations of SWMEDs in the same environment, just by using specific wavelengths of light. So, with the device itself established, we can now turn to that second big challenge: targeted delivery. This really gets to the central biological problem. How can a passively circulating electronic particle be guided to a precise location within the brain, across the blood-brain barrier, all without any kind of surgical intervention? The solution they came up with is a biohybrid approach. The strategy is to literally attach the SWEDs to monocytes, leveraging these cells' natural ability to hunt down inflammation and move out of blood vessels into tissue. As the slide outlines, this is done using copper-free click chemistry, which forms a very stable covalent bond between the SWEDs and the cell surface. The resulting cell electronics hybrids are then purified from all the unconjugated parts. After that conjugation step, the hybrid population was isolated using fluorescence-activated cell sorting, or FACS. This process yielded a population with a purity of 92.4%, and achieving such a high purity is really essential for the in vivo studies, both to ensure that the observed effects are actually due to the hybrids and to minimize any potential off-target effects. So to test this autonomous implantation in a living animal, a target had to be created. They did this by inducing focal neuroinflammation using a stereotactic injection of lipopolysaccharide, or LPS, into a specific brain region. 6 hours after that, the purified cell electronics hybrids were injected intravenously. Then, 72 hours later, the brain tissue was harvested for analysis. And the post-mortem confocal imaging confirmed that the targeting strategy worked. This image clearly shows the accumulation of these cell electronics hybrids, specifically within that inflamed brain region. This demonstrates that the hybrids successfully traveled through the circulatory system, crossed the blood-brain barrier, and homed in on that pre-established inflammatory target. Now, of course, 2 critical control experiments were performed to really validate the mechanism. When they injected SWEDs alone without the monocyte carrier, there was no brain implantation. And similarly, when they injected the complete hybrids into healthy mice that didn't have neuroinflammation, there was also no brain implantation. These controls confirm that you need both the cellular carrier and the inflammatory signal for this targeted delivery to work. To get a handle on just how many devices actually made it to the target, the researchers used a technique called inductively coupled plasma mass spectrometry to measure the titanium concentration in the brain tissue. Based on the known mass of titanium in each SWED, they calculated an average of approximately 14,000 devices successfully implanted per animal. This gives a solid quantitative measure of the delivery efficiency. All right, so the devices are in place. The next question is, are they actually functionally active once they get there? To test their function, 72 hours after the hybrid administration, the implanted SWEDs were wirelessly activated using transcranial illumination with near-infrared light. Then, 90 minutes after that stimulation, the brains were analyzed using immunohistochemistry for a protein called CFOS, which is a widely used marker for recent neuronal activation. The quantitative analysis of CFOS expression gave a very clear result. As this chart shows, the group that received both the hybrids and the optical stimulation showed a statistically significant, almost 3-fold increase in CFOS-positive cells compared to the control groups. This demonstrates that the neuronal activation was dependent on both the presence of the SWEDs and their wireless actuation. When they analyzed the spatial pattern of this activation, they found a very high degree of precision. This plot shows that the density of those activated cells drops off sharply to baseline levels within just tens of micrometers of the implantation zone. This indicates a neuromodulation precision of about 30 micrometers, a critical feature if you want to selectively target specific neural microcircuits. To get direct evidence of neuronal modulation, they also performed in vivo single-unit electrophysiology, and these recordings confirmed CFOS results. They showed that a subset of recorded neurons had a significant increase in their firing rate that was time-locked to the optical stimulation pulses. Further analysis of the spike timing revealed a high degree of temporal precision, which provides direct evidence of SWEDs-mediated neuronal control. Okay, let's move on to the final experimental section, which addresses the critical question of biocompatibility and safety. A pretty comprehensive safety assessment was conducted. As this table summarizes, behavioral tests for things like locomotor activity and cognitive function didn't reveal any deficits in the animals that received the hybrids. Furthermore, they analyzed complete blood counts, serum chemistry, and did histological exams of all the major organs, and found no evidence of systemic toxicity or any adverse effects. Now, to assess the innate immunogenicity of the devices inside the brain itself, SWEDs were injected directly into the cortex. The subsequent analysis of microglial and astrocyte activation markers, IBA1 and GFAP, showed no significant difference compared to a sham injection of saline. This suggests the SWED materials themselves don't provoke a significant neuroinflammatory response beyond the physical delivery procedure. The long-term stability of the devices was also evaluated. After direct intracranial injection, the SWEDs were observed to be stable for up to 6 months. And over this entire period, there was no detectable decrease in the number of devices, nor was there any histological evidence of material degradation or local cytotoxicity. This indicates a high degree of durability and material biocompatibility. And finally, they investigated the fate of the hybrids that don't actually implant in the brain. Using in vivo imaging of fluorescently labeled hybrids, they showed that the systemic signal returns to baseline levels over about 10 days. This indicates that any non-targeted hybrids are successfully cleared from circulation by the body's own endogenous clearance mechanisms. So, to wrap things up, let's summarize the primary achievements of this work and discuss some of the potential future directions for this technology. In summary, this study really provides the first proof of concept for the autonomous, non-surgical implantation of bioelectronic devices in the brain. The work details the engineering of functional subcellular devices, it demonstrates their capacity for wirelessly controlled neuromodulation with high spatial precision, and it establishes a comprehensive profile of their biocompatibility in vivo. The authors themselves frame this work as a foundational step. By successfully fusing electronic function with the inherent targeting capabilities of living cells, they've established a new potential paradigm for creating self-implanting bioelectronic systems, basically shifting the burden of delivery from the surgeon to the biology itself. Looking forward, several future directions seem apparent. Research could focus on increasing the implantation efficiency, maybe through cell engineering. You could develop new device materials for either chronic stability or for controlled bioresorption, and you could adapt the platform to other pathologies by using different cell types that have distinct biological targeting abilities. Furthermore, integrating sensing modalities could one day enable closed-loop systems for both diagnostics and therapy. Ultimately, this technology opens up a significant new research vector. It prompts us to consider how such autonomous cell-guided electronic systems might reshape future diagnostic and therapeutic strategies, not just for neurological disorders, but potentially for any systemic disease that's characterized by a localized, targetable biological signature.